Hello everyone. My name is Adeeba and I'm from Katihar Medical College. And in the previous session, we left pteridophytes. We were doing the important discussion on pteridophyte, which comes under the five classification, the five classes of the plant kingdom. The first one was algae, the second is the bryophyte, then pteridophyte, the gymnosperm, and then the angiosperm. So we were studying about pteridophytes. Important points of pteridophytes and its economical importance, its ecological importance, we have already covered in the previous session. Some of the ecological importance which we studied was, first of all, that the pteridophytes, they are sporophytic animals. The sporophytic plants. The main plant body is 2N and they give rise to spores. These spores, when germinate, they form gametophytic plant body by the production of gametes, and Antheridia and Archegonia are born on the protonema, and then the fertilization takes place and a new plant body is formed. Whereas when we talk about the importance of pteridophytes, they are mainly an ornamental plant because of their beautiful leaves, broad and different shapes of leaves are present in the pteridophytes, which is the main reason why they are usually used as an ornamental plant. And they are the first plants. Whenever a forest fire takes place, they are the first plant to colonize that part because they have rhizome. They have roots, which is actually rhizome. Whenever the surface plant gets destroyed due to fire, these rhizomes they give rise to stem and the new plant body is formed. Now, the third point was that they can also be used in transportation. Sometimes they help in transporting some or the other thing from one place to another. And the third is that they are also used in some medicinal purposes as well. So there was not a lot of uses for pteridophyte, but yes, still some were present, out of which I have stated three to four of them. Now, pteridophytes, they are further classified into four classes. The first one was Psilopsida, the second is Lycopsida, third Phenopsida, and the fourth is Pteropsida. So we have looked into Psilopsida in the previous session, but we'll just do a quick revision of it. Psilopsida, it is also called as piscine. The plant body is rootless, sporophyte, differentiated into a subterranean rhizome and an aerial shoot. So there is a subterranean rhizome which is present in the underground and an aerial shoot which we can see. Branching is dichotomous in both subterranean rhizome and aerial shoot. So the branching is very profusely present. They are very highly branched. Scale-like or leaf-like appendages are borne spirally on the aerial shoot. So on the aerial shoot, there are some scale-like or leaf-like structure which are borne. Sporangia develop over the aerial stem terminally or axillary. So sporangia, they develop terminally or the axillary. Now they are thick-walled and homosporous. Antherozoids are multiflagellate. Why the antherozoids are multiflagellate? Because it will help in the movement of antherozoids to the archegonium, which would aid in fertilization. Examples is Rhinia and Xylotum. The Rhinia is the one which is extinct, but Xylotum it is seen around us. Now, Xylotum. So Xylotum it grows on soil and grows as an epiphyte. What is epiphyte? Epiphyte are the type of plant which grow on another plant and they extract the nutrition from the other plant. Now, plant body is differentiated into rhizome and an aerial stem. We can see that the plant body is differentiated into rhizome and then aerial stem. This is rhizome and this is aerial stem. Now, the roots are absent. There is absence of root, but rhizoids are present. Rhizome is non-green, branched, and subterranean, bearing stem and filamentous rhizoids. So this Rhizome, this rhizome which is present, it is first of all it is non-green, it is branched and it is subterranean. It means that it is present inside the soil and it bears stem and filamentous rhizoids. So the stem is green and dichotomously branched. And the stem which is present, it is green and it is branched. Okay, green and branched. It bears number of scales. So some scaly structures or leaf-like structure are present. It reproduces vegetatively by gametes or asexually by spores. So it can reproduce vegetatively by the production of gamma. We have studied about gamma in bryophytes as well, but in this case the gamma are different. And gamma are small multicellular structure which are born over the rhizome. The gamma cup which was present in bryophyte, it was born on the thyroid surface of the body. But here the gamma that is present, it is born on the rhizome. Gamma is present. Okay. Now spores develop meiotically in short-stalked trilobed synangia or compound sporangia. So the spores which develop, they develop meiotically in a short-stalked trilobed synangia. There is a trilobed synangia, or we can also call it as a compound sporangia. Why compound sporangia term is used? Because there usually in the sporangia there is only one lobe present inside which the spores are produced. But in this case, it is called as synangia because there are three lobes present. It is trilobe. Multiple lobe, that is why it is called as synangia. Syn term is used for together. When two or more things are joined together and then they perform the function, it is termed as synangia. So the synangium develops in the axil of the bilobed foliar appendage. So on the foliar appendages, that means the leafy structure, on that part, the synangium develops and it will give rise to production of spores. Now these spores, they are dispersed by air. On a suitable substratum, each spore germinates to form a colorless prothallus. So when a suitable substratum is present, each spore will germinate to form a colorless prothallus. Now this prothallus, it is free living. It has an endophytic fungus that aids it in nutrition. Prothallus multiplies vegetatively with the help of gamma. So prothallus can also multiply vegetatively by the help of gamma, and when the prothallus it grows, it will give rise to male and female sex organs that are antheridia and archegonia. So it reproduces sexually by the formation of antheridia and archegonia. When antheridia and archegonia are produced, inside the antheridia, antheridia are produced, and inside the archegonia, which is a flask-shaped structure, eggs are produced. And these antherozoids which are produced, they are biflagellated. For what? They are biflagellated so that they can swim to the archegonia and fertilization can take place. So on fertilization, embryo is formed which grows to produce a new sporophytic plant body. So the life cycle of Tylotum or even any other pteridophyte, it is very easily understandable. And how that happens is that the sporophytic plant, this sporophytic plant, it will give rise to spores. And this spores are produced by meiotic division. These spores, when they germinate, give rise to prothallus. And this prothallus will have male, female, and male sex organs, antheridia and archegonia. And when the gametes fuse, it will happen when the gametes fuse, it will give rise to production of zygote, and this zygote will again form sporophytic plant body. Okay, now Lycopsida. Lycopsida is a very primitive vascular plant. The plant body is differentiated into roots, stem, and leaf. Leaves are small with a single unbranched vein. So there, the leaves they first of all they are very small, and the veins they are not branched. There is a single unbranched vein. Sporangia develop in the axil of the sporophylls. So these sporophylls which are present at the axil on which the sporangia is formed. The sporophylls form a compact strobili, so they are called as cones as well. The examples are Lycopodium and Selaginella. So this is the figure of Selaginella. We can see that root, stem, and leaf all are present. The roots are present, stem is present, and leaves are present. Leaves are small and they are not branched. The venation in it is not branched. Selaginella. So Selaginella is called as club moss or spike moss. The Selaginella grows in damp and poorly illuminated forest floors, which means that Selaginella grows in damp and shady places. Like all the other pteridophytes and bryophytes also grows in damp and poorly illuminated forest floors. A few species occur on moist hillsides. So some of the species might be occurring in the hilly areas as well. A few species grow in xerophytic condition as well. Xerophytic condition is the condition which is found in deserts where the water is very less. So when plants they grow in the desert areas, they are called as xerophytes, and the condition is called as xerophytic condition. They roll up in brown balls during drought. So how they cope up with the loss of water which actually happens there in the deserts, they roll themselves. They roll up themselves into a brown balls. Some brown balls, if you please see in desert areas, they are usually selaginella. It is called as cespitous habitat. On being moistened, the dried plants straighten and turn green. So when this rolled plant is provided with some water, it will again get back to its normal shape and size, and it will straighten out and it will turn green. Therefore, they are called as resurrection plants. Resurrection plants. Resurrection is the term which is used when a person when a person dies and is it starts living again. Then it is called as resurrection. So they are called as resurrection plants because we first assume that the plant has died because it has dried up and it has converted into a shape like a ball. Whenever water is provided to the particular plant, it gets back to its original shape and size. We have resurrected the plant, given life to it back again. So it is called as resurrection. The examples are Selaginella lepidophylla and Selaginella bryopteris. Vegetative reproduction occurs by fragmentation. We very well know that the vegetative reproduction, the spore and the gamete formation doesn't take place and it takes place by the part of the plant which gets germinated nearby in the soil and a new plant body grows. So the vegetative reproduction occurs by fragmentation, tubers and bulbils. Tubers are formed and bulbils are formed by which the vegetative reproduction takes place. The Laginella is heterosporous as it produces types of spores. Heterosporous, why? Because it produces two types of spores. So the one spore will be all microspore and the other one will be macrospore. Megasporangium is shortly stalked and produces megaspores. So the megasporangium, which is shortly stalked, it will produce Megaspores, while as microsporangium has the longest stalk, reniform or globular in form and produces number of small microspores. Why the megasporangium is shortly stalked and the microsporangium is longer stalked? Because the megasporangium is shortly stalked because it bears only one egg, and the megasporangium or the egg it does not have to travel, so it can remain on the plant, attached to the plant body, and it will remain nearby the plant. But the microsporangium, the microsporangium will give rise to production of microspores, and these microspores have to disperse. They have to germinate, which is the reason why the microspores they are located a bit away from the plant body so that they can help in germination. Okay? A microsporangium has the longest stalk, reniform or globular in form, produces a number of small microspores. The sporangia are born on the strobila. Sporangia, they are born on strobila, which we called as compact stones. Those were termed as strobila. We have seen a diagram in which there was strobila present on the top of the stem-like structure. Now, each microspore grows when the microspore it germinates. It will give rise to male gametophyte, which has a single prothalleel cell and an antheridium with 120 sperm cells or androcytes. So this microspore, when germinated, will give rise to male gametophytic body, and this gametophytic body will have a single prothalleel cell. There is a prothalleel cell and an antheridium. Antheridium inside which the androcyte or sperm cells are present. And how many androcyte or sperm cells it is bearing? Around 120 cells. The megaspore will grow into female gametophyte, and the female gametophyte has an apical cushion, a secondary prothallus, or endosperm with food-laden multinucleate cells. The thing that they're talking about, endosperm with food-laden multinucleate cells, it is actually nucellus. When we study angiosperms, nucellus is present, which helps the survival of an embryo. They will help in the nucellus cells will provide nutrition to the embryo, which will help in its survival throughout the dormant phase. So the female gametophyte will give rise to the megaspore will give rise to female gametophyte, and this mature female gametophyte it has an apical cushion and a secondary prothallus, and secondary prothallus is actually food laden and multinucleate cells, food laden with multinucleate cells. Apical cushion develops archegonia and rhizode. So the apical cushion which was present on the surface of a female gametophyte, it will develop archegonia and rhizode. After fertilization, embryo is formed and embryo has a food for absorption of food. So the embryo which is formed, it is actually independent. It grows out of the female gametophyte into a sporophyte, and because it is born on the plant body, it will grow out from the female gametophyte into a new plant that is a sporophyte. So each microspore which is present will give rise to a male gametophyte, and a single male gametophyte will have single prothalleel cell and an antheridium, and inside the antheridium, antherospores, androcytes or sperm cells are produced, which are 120 in number. Whereas megaspore, it will give rise to female gametophyte. This female gametophyte will have some peculiar structures such as cushion, secondary prothallus, and secondary prothallus is actually an aggregate of multinucleate cell which helps in nutrition. Now, the apical cushion which is present, it will develop into archegonia and rhizoid, which after fertilization, when embryo is produced, this embryo has a food for absorption of food and it will be present on the female gametophyte. And after the detachment, it will grow into another sporophytic plant. Now, the life history of Selaginella. So, history of Selaginella, up till now, we have understood that it is diplohaplontic. Diplotic, diploid cells or sporophytic cells in the first half of the life, and then in the second half, it is gametophytic. So, life history of Selaginella is diplohaplontic. It shows heteromorphic alternation of generation. We have studied about the alternation of generation, and we have also studied that it is of two types: isomorphic and heteromorphic. Isomorphic alternation of generation is said to be present the two when the two generations, the gametophytic and the sporophytic generation, or the diploid and the haploid generation, they both resemble each other in structure, size, shape. But whenever the gametophytic and the sporophytic generation resemble each other, there is some kind or the other kind of difference between them, then it is called as then it is termed as in heteromorphic. Hetero means different and morphic means morphology. If they are different in morphology, they are termed as heteromorphic. Isomorphic when the morphology of the gametophytic and the sporophytic plant body is same. The seed habit of the Selaginella. Selaginella shows several adaptations to produce a seed. Actually, if the term the presence of seed habitat that is used for pteridophytes. The production of seed started from gymnosperms and angiosperm. The seed is present in both gymnosperm and angiosperm, but the beginning of the formation of seed is termed as seed habitat, and it starts from pteridophytes. So the vascular plants, the plants which possess the vascular system for the first time in the whole plant kingdom, they were pteridophytes. The second thing that they show a very important structure, very important step in the evolution, that is the presence of seed habitat. They are actually preparing the whole of the plant kingdom which will come after them. The pteridophytes are the primitive one after which the gymnosperm and angiosperm came. They are preparing them for the retaining of seeds. So seed habitat, the formation of seed actually started from gymnosperms. They were preparing the body in order to retain the seed on them. So, seed habitat of Selaginella. Selaginella shows several adaptations to produce a seed. These are presence of heterospory. The first of all, the presence of heterospory is an important indication for the presence of seed habitat. The oogamous kind of reproduction is required in order to produce seed. First thing. The second is the formation of two types of gametophytes. Very obvious that two types of gametophytes should be produced. The first one should be male and the other one should be female. Male gametophyte and female gametophyte should be produced in order to prepare a seed. The dependence of gametophyte on parent plant for nourishment. Very important thing that the gametophyte should depend on the sporophytic plant body, and when the sporophytic plant body will let the gametophytic plant to depend upon itself, it is actually helping the gametophytic plant body to grow. It will retain the seed on the sporophytic plant body. Okay. Now, presence of single megaspore, very important that when a single megaspore will be present in the female gametophyte, the sperms will fertilize the single egg and a seed is produced. If multiple ovum are present, then the production of seed might not take place that easily. Retention of megaspores within the megasporangium. Very obvious that when the megaspores will be retained in a megasporangium, then only the seed habitat can take place. Then only the sporophytic body will provide nourishment to the seed. If they are present away from the body, how will it come back to the plant for its nourishment? It is not possible. Sort of pollination and growth of male gametophyte near female gametophyte. So, a sort of pollination should be present first of all, so that the male gametophyte can travel to the female gametophyte easily. It can be through water, or it can be through air, or by any means of transport which the plant might take. And the third is the presence of male gametophyte near the female gametophyte, so that the male gametophyte do not have to travel that far for fertilization. Now, retention of megagametophyte, and finally the development of seed. When this megagametophyte will retain on the plant body, then the seed production will be there. So these are the some points which were present in pteridophytes which helped in the production of seeds. Oh, the Stenopsida, which are horsetails. So stem is differentiated into nodes and internodes. The nodes and internodes are present, and leaves are borne on nodes, sporangia on the sporangiopores, which form a compact cone. So the body looks something like this. The leaves are borne, and where the leaves are borne, it is called as node. The space between the two nodes, it is called as internode, and on top, the compact cones are present, which are called as tobuli. These compact stones are actually sporangiophores. The sporangium are present inside which the spores will be produced, and where the leaves are borne, they are node, and the space between the two nodes, it is termed as internode. Example is equisetum. We can very clearly see in the diagram that this is root, and where the leaves are produced, it is termed as node. This is node, and the space between the two nodes, it is internode. Trobilus is present, which forms a conical structure. Now, equisetum, it is also called as horsetail or scouring rushes, is a remnant. Fossitum is the remnant of large group of pteridophyte, and it is called as living fossil. So these are termed as living fossil. The plants are popularly called as horsetails because of the whorled branches and compact terminal strobili. These are popularly called as horsetails. Why? Because of the presence of branched and compact terminal strobili. They are also called as scouring brushes because of their use in scouring dishes due to roughness of the surface. Scouring, we have learned the term scouring before. That scouring, it is used in polishing or washing dishes or polishing some materials with the help of the plant. So equisetum is used for scouring because this plant, it is rough, and the roughness will help in polishing the materials. So equisetum, they are called as horsetails or scouring rushes. Postum is remnant of large group of pteridophytes, which is living fossil, and they are popularly called as horsetails because of the whorled pattern, whorled branches, and compact terminal strobili. And they are also called scouring brushes because they are used in scouring dishes due to roughness of the surface. Now, equisetum, it grows along the banks of rivers in sandy or swampy soils. The plant body is differentiated into stem, root, and leaf. We have seen there was a stem-like structure, stem and root and leafy structures. Okay, so the plant body is differentiated into stem, root, and leaves. Stem has two parts, the aerial and the underground. So this is an underground. There is an underground stem. Okay, underground stem, it is called as rhizome. It is highly branched. Nodes and internodes are distinct. Nodes and internodes are very clearly visible. The nodes internodes are hollow while nodes are solid. So the nodes will be solid and the internodes they will be hollow. Leaves are microphyllous. We can see that the leaves which are present in the ecosystem they are very small. Microphyllous leaves. The leaves are microphyllous, simple and scale-like, arising like whorls from each node. So they are scale-like and they arise in whorls from each node. In few species, the plants bear two types of aerial branches, green sterile and brown fertile. So there may be in some species, the branches can be of two types. The first one is green and sterile, and the other one is brown and fertile. Fertile branches bear strobili or cones at the tip which produce spores. The spores are homosporous, which germinate to produce independent cushion-like gametophytes. These spores, when they germinate, they give rise to an independent cushion-like gametophyte. Vegetative reproduction takes place by fragmentation and the formation of tubers. Vegetative reproduction can be by fragmentation or by the formation of tubers. Now, Pteropsida or Filicopsida, which is called as ferns. Plant body is differentiated into root, stem, and leaf, like all the other we have discussed. Leaves are large and pinnately compound. Sporangia develop on the ventral surface into sori. So, sporangia they will develop into the sporangia they will develop on the ventral surface, which is called as sori or sorus. In the diagram, we can see that there is first of all the division is present, root, stem, and leaf, and the leaves they are pinnately compound. The pinnately compound leaves look something like this. There is one rachis, okay, and these are the leaflets, not a leaf. The whole is termed as a leaf, and these small leaflet structure they are leaflets. Example of which are Dryopteris, teres, and Radiantum. Now, Cryopteris fern. Cryopteris is commonly known as a shield fern. Its plant body is differentiated into root, stem, and leaf, and the stem is rhizome. So the stem is actually a rhizome. It grows obliquely upwards through the soil. The rhizome is covered with the persistent basis of dead leaves. So the rhizome it will be covered with persistent bases of dead leaves. The stem does not bear aerial shoots. The stem will not have aerial shoots and is usually unbranched. You can see the stem, the only one stem is present. There is no branching. The roots are adventitious. They arise as small and wiry structures. You can see the wiry structure present from the lower surface of rhizome and fix it into the substratum. So the roots are usually adventitious and they arise as small wiry structures from the lower surface of rhizome, and these wiry structures help in the fixation of the plant to the substratum. So Dryopteris, it is what it is commonly called as. It is commonly called as a shield fern, and its plant body it is differentiated into root, stem, and leaf like all the other pteridophytes. The stem is rhizome and it grows obliquely upward. It will grow like an oblique structure growing upward. The stem is not the stem is not branched, first of all, and second of all, the stem is mainly rhizome, which is covered by the debris of dead leaves. Third, that the roots which are present, the roots are actually coiled structures, and due to this coiling, the roots help in the attachment of the plant body to the substratum. The leaves they are large and arise from the upper surface of the rhizome, and they form most conspicuous portion of the plant. So they are the most conspicuous portion of the plant. and they are petiolate and pinnately compound. Petiole is a structure which this is a petiole which helps in the attachment of the leaf to the stem. So there is a petiole present and pinnately compound leaves which are the leaves in which incisions are present due to which they represent as leaflets. The young leaves have a circinate they are rolled from the apex downwards. So we have very commonly seen this kind of pattern in the newly growing structures of plants that the newly growing leaf or even the newly formed flowers they form a bud so that they can protect the very fresh and young cells from damage. And this is the same reason why the leaves they are rolled. They are rolled at the end, at the bases, so that they can protect themselves from the outer environment. And when they'll grow, it will give rise to a proper leaf-like structure. The venation is bucket and the leaflets are called as spinnet. So the leaflets which are produced due to the incisions on the leaf, they are either called as leaflets or they can also be called as pinna, which in turn have pinnules. So they even have pinnules. The younger parts of the rhizome, petiole, and leaf are covered with brown hair-like structure called ramenta. We have discussed the term ramenta, which is a hairy-like structure present on the surface of the newly growing plant or even on the stem or petiole, so that it, the presence of this will help the plant to protect itself from desiccation. The ramenta, it is protective in function. Okay? So the description about the leaves says that they are actually large and they arise from the upper surface of the rhizome. Why the upper surface of the rhizome? Because the stem, it is mainly rhizome and they form most conspicuous portion of the plant. They are petiolate and they are pinnately compound. Petiolate means that they bear a petiole and petiole is a structure which helps in the attachment of the leaf to the stem. And pinnate means that they have incisions on the leaf due to which leaf-like structure leaflets are formed. The third is that the young leaves they get rolled. They get rolled near to the body of the plant. The venation is purcate and the leaflets are called as pinnae. The small leaflets which are present, they are actually pinnae, which in turn they can also be further divided into pinnules. The younger part of the rhizome petiole and leaves are covered with brown hair-like structure, the ramenta. So the ramenta is present, browny hair-like structure, and the ramenta is protective in function. Now reproduction. Reproduction may be vegetative, asexual, and sexual. We know about it. Vegetative reproduction it takes place by the fragmentation of rhizome and by the adventitious roots. The adventitious roots. Asexual reproduction, ferns reproduce asexually by spore formation. Very obvious that the asexual reproduction take place by spore formation. They are buds or bulbils. So buds or bulbils can also be present. Homospores and spores are known as meiospores, produced by meiosis, and spores are produced in spore sacs or sporangia. They are aggregated in groups called as sori on the undersurface of the sporophylls or leaflets. So these sporangia they are aggregated in a group and that group is termed as sori and this sori is born on the undersurface of the leaf. This sori occur in two rows, one on either side of the midrib and one like there is this leaf and there is midrib. So one will be present on one side and the other one on the other side. Each sore is protected by a membranous covering called as inducium. So this sori, what is actually sori? Sori is an aggregate of the sporangium Second, it is present and in the undersurface of the lateral or the lateral surface of the leaf. The third, they are present in two rows, one on the each side of the midrib. Fourth point, that they have a protective membranous covering over them, which is called as inducing. Now, the spores are minute. They are very minute, haploid, and dark brown structure surrounded by a thick, dark, and rugged wall. So, these spores they are very minute and they are haploid. And spores will be haploid. Why? Because they are produced by meiosis. These spores, which are minute, haploid, and dark brown, they are surrounded by a thick and dark rugged wall formed by exine and intine. Now, the spores, the exine and intine, they are covered by exine and intine, which are very thick structures. These structures actually help in storage of, or even they prevent the cell membranes of the spores to get destroyed from any of the environment, be it water or wind or anything, drought, because these are very hard structures which cover the spores and help in their survival through the extremes of the condition. On getting favorable conditions, the spore germinate into a short green gametophyte, which is called as prothallus. So the first step was that the production of spores. How the spores were produced? We know that the sporophytic plant body, by the help of meiosis, it produced spores. And these spores have a peculiar structure, that these spores are very minute, haploid, and they are dark brown structures which are surrounded by thick, dark, and rugged wall, which is formed of exine and intine. Now, whenever the favorable condition arrives, these they germinate, and when they germinate, they give rise to a short green gametophytic body, which is called as prothallus. And now we will see what prothallus gives rise to. Prothallus, the mature prothallus is heart-shaped. It is about 4 to 4 inch in size and is attached to the soil by a numerous unicellular brownish rhizoids. You can see that this is a heart-shaped prothallus with the presence of rhizoids. The prothallus is monoecious and bear both archegonia and antheridia on its ventral surface. So it will bear both. The archegonia is present as well as antheridia on the ventral surface. Ventral surface means the anterior portion, which we can see. Now, the sexual reproduction. How will the sexual reproduction take place? The antheridia and the archegonia will bear the sex organs. These are the sex organs, and it will bear egg and the sperms, and when they fuse, the sexual reproduction will take place. So the third step which we talk about is sexual reproduction. Antheridia produce flagellate antherozoids or spermatozoids, are which reach archegonia by swimming through water. One of them fuses with the egg cell, and the diploid zygote thus formed is called as oospore. Okay? Now the diploid oospore marks the beginning of the sporophytic generation. Very obvious that this zygote which is formed, which is diploid, it is called as oospore, and oospore will give rise to a gametophytic plant body, the sporophytic plant body. It secretes a wall around it and undergoes segmentation by ordinary mitotic division. The sporophytic plant body, all this oospore will divide mitotically, and mitotic divisions will take place and a new young embryo is formed. The foot of the young embryo remains in close contact with the prothallus and absorbs food for the young with the sporophyte. Okay. So when the fertilization takes place, oospore is produced, and this oospore will give rise to sporophytic plant body. When the oospore is produced, the foot of the young embryo it remains in close contact with the it remains in close contact with the prothallus, and it will absorb food for the young by the help of the sporophyte, and this sporophyte will give rise to another plant body. As we can see in the picture, The prothallus, the gametophytic plant body, by the help of spores, germination takes place and Antheridium and Archegonia are produced, oospore and antherozoids are produced, oosphere and antherozoid are produced with the production of oospore. Embryo produced and the sporophyte is produced. So this diagrammatic representation or graphical representation is actually the representation of the life cycle of pteridophyte. How pteridophytes reproduce, what are the stages, the diplontic and haplontic stages of the plant body, and the gametophytic and the sporophytic stages. Okay. So the life cycle of pteridophyte exhibits heteromorphic alternation of generation. Heteromorphic alternation of generation. Its sporophyte is more conspicuous and it is called a spawn plant. It is differentiated into root, stem, and leaf. So the sporophyte is more conspicuous. We can very clearly see it, and it is called a spawn plant because it is differentiated into root, stem, and leaf. Now, very obvious that this sporophyte will be a spores by reduction division or by meiosis in the spore mother cell. The spore mother cells are the last structures of sporophytic generation. So, if we have been asked that what are the last generation of the sporophytic generation, last structure, so they are the spore mother cells. The spores initiate a gametophytic generation. Now, these spores will give rise to gametophytic generation. Haploid spore germinate to form prothallus, and it is the gametophyte and reproduces sexually by the formation of gametes. Obviously, the after germination of the spores, the gametophytic plant body is produced, which will give rise to production of gametes. The sperm and the egg are the last structure of the gametophytic phase. So, the sperm and the egg are the last structure of gametophytic phase, after which, when the fertilization takes place and zygote is formed, again the restoration of the sporophytic plant body takes place. So, this type of life cycle is called as haplodiplontic life cycle because there is one stage is haploid and the other one is diploid. So we have completed pteridophytes in a very great detail, and in the next class we will study about seed and the how after pteridophyte from gymnosperms seed formation takes place and then gymnosperms. Thank you so much.